Hello everyone, I am Dr. Sirajul Faisi. I welcome you in my YouTube channel. Today in this video, we are going to learn amperometric titrations. You know friend, this topic is very important for JAM, NET and GATE examinations. So let us start to learn amperometric titration. You know friend, polarographic methods has been adopted to detect end point of titration. Polarographic methods has been adopted to detect end point of titration. You know the end point. From Ikovic equations is directly proportional to, or ID is directly proportional to concentration of reducible species. Graph of diffusion current versus volume of titrant added allows the end point of titration to be determined. What does it mean? Titration procedure. You can understand first titration. Known volume of experimental solution is placed in polarographic cell. Suitable supporting electrolyte, usually potassium chloride, is added. DME is inserted in cell and it acts as cathode. SCE electrode used as reference electrode. N2 is bubbled for 5 minutes. Removal of dissolved O2. Applied voltage depends upon reducible species and ID is measured. Titrant is added through microburette. So, shape of titrations curve depends upon the species reduced at DME and applied potential. They are classified in 3 types. My friend, these 3 points is very important. Titrant is reducible, but other species are not. Titrant is reducible, but other species are not. Both titrant and titrant are reducible, but product is not. Just keep in your mind these 3 important points. Now, titrant is reducible, but other species are not. Titrant containing reducible species such as lead plus 2 is titrated with a non-reducible species such as sulfate or oxalate ions, which is also precipitating agent. Here sulfate 2 plus, not 2 minus. It is the mistake. Diffusion current decreases linearly with increasing volume of titrant added. So current versus volume of titrant, you will get such type of graph. Now the second condition. Titrant is reducible, but other species are not. Titrant containing non-reducible species such as SO4 2 minus, not 2 plus, SO4 2 minus or oxalate ions with a Pb2 plus or barium 2 plus reducible species. Titrations proceeds, the current remains steadily till the end point since no species is reduced. So my friend, after equivalent point, diffusion current increases linearly with increasing volume of titrant added. So, you will get current versus volume of titrant, this type of graph. Both titrant and titrant are reducible, but product is not. Just keep in your mind. A titrant containing reducible species such as Pb2 is titrated with reducible species such as Cr2O7 2- ion. In this diffusion, current decreases first, since titrant contains reducible species till end point, and then increases with additions of titrant, which is also reducible. So V-shaped graph shown in figure. So current versus volume of titrant, you will get this type of, which is over here. Now, what is the advantage of empireumatic titrations? Method is quite accurate and end point determined by means of a graph. The electrode surface continuously renewed. Consequently, current are reducible, and this is useful in precipitation titration. Mercury exhibits a high over voltage for evolution of hydrogen. This used to yield irreversible polarizographic waves. My friend, it gives more accurate results for diluted solution as compared to other methods. Reading near end point are not important in this method. So what is the disadvantage of this technique? Considerable time required to remove dissolved oxygen before and during titrations. The titration cannot carried out as potential more negative than 2 volt since H2B evolved. So co-precipitations can give inaccurate results. So my friends, I hope this video will be beneficial for you. I would suggest, especially for JAM, NET and GATE exam, me, who is struggling for qualifying such type of exams, and national level other competitive exam of chemistry. So if you like this video, then subscribe my channel and share it in your friend circle. Thanks for watching.